Okay. பயங்கர thin keyboard. Okay, thanks. யாரோ கேட்டாதான் நான் பேசுவேன். வெயிட்.
Question raise பண்ணு. 20 seconds later.
Oh, okay.
What is it?
இப்போ நான் பேசலாமா? அப்போ 2-3 கொஸ்டின் தான் இருக்கு. Okay. So good afternoon, students. I hope you are enjoying these developmental biology lectures. So some of you have sent questions by the Google form. So I have a few with me. So I'll go ahead and answer one by one. So the first question is from Arunima. The question is, what do you mean by embryo? Okay, so embryo is the structure that is formed by the fusion of oocyte and sperm. Okay, so that when the two haploid nuclei fuse and form a diploid nucleus and a cell, we call that as zygote. So that's the earliest stage of the embryo. And then that embryo undergoes cleavages, forming 2 cells, 4 cells, and so on. So the entire stage, starting from fertilization, the first diploid nucleus, all the way to the completion of development, when we talk about at least mammals, up to the birthing of the new offspring, that entire process of embryogenesis, that entire state, we will call it as an embryo. Okay, so that is an embryo. And another question from Sanjay. This question is human placental cell types. Okay, so human placenta actually has 2 main contributions. One from the maternal side, so that is primarily derived from the uterine wall. So the uterine wall cells are the ones that are modified into one part of the placental structure. So we call this as the deciduum. Okay, it primarily provides nourishment, and it also acts to clear away the used products and materials to be disposed of, for example carbon dioxide, from the embryo. And the other part of the placenta, made from the embryonic structures, essentially extra embryonic structures, derived from trophectoderm, and that portion is called chorion. So the placenta has 2 main parts. One is contributed by the mother and another one by the embryo. So the maternal thing derives from the uterine wall tissue, and the embryonic side comes from the extra embryonic tissue. That is, when the embryo starts to divide, a few cells initially are set apart, called the inner cell mass. That is the one from which the actual embryo will be formed. The remaining cells are going to form the extra embryonic tissues, which are primarily provide a support and link between the mother and the fetus. And the third question that I have comes from Dr. Radha Dayanidhi. She's a faculty, and her question is, can you elaborate on the explanation related to two families of proteins that maintain the memory of transcriptional state throughout adulthood? I guess this question pertains to our discussion on Drosophila, embryonic patterning, and the gene regulation involved in that. So there we saw a hierarchy of genes acting, starting from the maternal effect genes, then gap genes, pair rule genes, and homeotic genes, and so on. And once these transcription factors activate or suppress certain gene expression, that needs to be maintained, because these initial master regulators, their expression is dynamic. They are not going to be continuously be expressed in the same way. So the activated or repressed state needs to be maintained, and that is primarily done by the proteins belonging to two families. One is the trithorax and another one is the polycomb group. So these proteins essentially bind to the chromatin, and one of the mechanisms by which they activate or mechanisms by which they maintain the activated or the repressed state is by modifying the chromatin itself. Primarily this is the methylation if it is repression, or acetylation if it is activation of the histones. Okay, so that is how they maintain the chromatin architecture, and they remain bound to the chromatin also. So that is what this trithorax and polycomb group family proteins do. So the main thing you want to remember here is the activated or suppressed state is maintained by histone modifications. With the modifications are acetylation and methylation of the histone residues. Okay, so histone proteins, the amino acid residues of the histones get methylated or acetylated. Okay, so normally deacetylation and methylation leads to repression, and removal of the methyl group and acetylation leads to activated state. So that's the other question I have. And someone is asking, any course available on biomimics in engineering? Very interesting topic, but unfortunately I don't offer that course. You need to look through the NPTEL's entire set of questions offered there and check whether they have a course like this. So right now I can't satisfactorily answer you this question, because I do not know whether they have a course on biomimics. Now I see some questions popping up in the chat box. So there is one question from Harshit Mishra. I have been having difficulty with making notes of the lectures. The slides have mostly pictures and not much of notes. So can there be notes provided separately apart from transcript? So essentially the transcript is the notes. Exactly whatever you need to know is what I normally elaborate in the lecture. And that entire thing is made available as transcript. If you want more than this, I certainly urge you to read the textbook by Scott Gilbert. Okay, so you can read 7th edition or any of the later editions. I'm sure many of you think listening to a lecture by a teacher helps learning. I agree, but at the same time, there is no substitute for reading a book. When you read a book, you get to know a lot more details than what one would explain in a 45 minutes to 1 hour lecture. That is one thing. Second, when you read a book, you can try to go at a pace, that is the speed of understanding, that is comfortable for you. You can frequently revisit. You can go to a previous paragraph. reading is an absolutely important requirement on any topic. That I just cannot exaggerate on this point. And I'm actually, as a teacher, not very pleased to see people not reading anymore. Okay, so you can't listen and read a transcript or read notes and think you have understood the topic. You definitely need to read a textbook on a given topic. Here, this Scott Gilbert is a wonderful textbook, very nicely explained in very simple, straight English with nice cartoons and diagrams. So please go through the book. So that is what I would answer for that. How do Chandrapal Singh, how do our cells know when to stop dividing? Okay, so that's an important question. We did talk about it at the very first lecture itself. One of the outstanding questions of developmental biology is how do our cells stop dividing? Okay, when to stop dividing. So remember, I said at that point, our body structure has formed through several rounds of cell division, but just one more additional division means you will be a monster like objective. Okay, so that means knowing when to stop dividing is absolutely crucial. So this is still being actively researched, but we have understood a lot of basics. Much of it is covered in cell biology classes, but I'll briefly answer to your curiosity here. So we have a heterodimeric protein. Okay, these are called cell division dependent. Cell cycle regulated proteins, often called cell division cycle proteins or CDC proteins. I said heterodimer, so one of the dimers here, one of the monomers here is a kinase that has the catalytic activity that phosphorylates proteins that are involved in cell cycle itself, cell division process itself. And this phosphorylation may inhibit or activate those proteins, and that is how cell cycle is regulated. And this kinase itself, its activity is regulated by the other monomer called cyclin. Okay, so therefore these are phosphorylating enzymes that are cyclin dependent. And the cyclin transcription, cyclin translation, mRNA translation, and cyclin protein stability, these are all tightly regulated by upstream activators that control the organ size. And at cellular level, these are the main molecules, cyclins and their corresponding kinases. Okay. So you have, like, for example, CDK, say cyclin dependent kinase 1, and that is regulated by a cyclin called cyclin B. So like that we have cyclins and cyclin dependent kinases. So this heterodimer is the main regulator of cell cycle. And the most of the time the CDKs, cyclin dependent kinases, their expression stability are not strictly regulated. They are pretty much present all the time, but their activity is regulated by the cyclins, and the cyclins availability is what is subject to regulation at all the levels: transcription, translation, as well as protein stability. So that is my brief answer to that question. So now we have more questions. Transcript is hard to navigate for preparation. Okay, so I'll get back to that issue after talking to the TAs, probably through your discussion forum. Okay, so I'll first need to talk to the TA to see what is the problem with the transcript. But in my opinion, after listening to the lecture, If you read the book, that is the best thing to do. If I were a student, that is what I would do. Without it, I will never be satisfied. Okay. So book has a lot more examples than what I mentioned in a class. So I would again reiterate the same, but then I'll try to discuss with the TAs to see why transcripts are hard to navigate. Please adjust camera ISO. It is flickering. Okay, that you are doing. One more question. So the Scott Gilbert book is quite costly. So can images of pages relevant to lectures be provided, or is there an online free PDF available? Well, you will have to prioritize the way you spend your money. I'm sure you do buy other stuff where you probably can prioritize and try to find ways to buy textbooks. This is what is very important for your future career. So you should prioritize and focus on the book, because it's like copying images by the book and providing in a public forum like NPTEL is illegal. So I will not be able to do that. Even for what is there in PowerPoint, I wrote to Scott Gilbert himself and got permission, and with that permission only we are providing you the PowerPoint slides, and PowerPoint slides have all the cartoons that you need. So you need to go to the textbook primarily for the text itself. So you may be able to find a cheaper PDF version on the internet that you need to navigate to their website and check. And I'm sure, I think now, originally it was published by an American publisher, Senior Associates, but now it is available through Oxford publishers, and they are in India, and they probably have an Indian version which must be cheaper. So you need to check that out by searching on the internet and see. Because we will not be able to provide you scanned copies, and that is actually illegal. It's not allowed. And I would not encourage you to download illegal versions from the web either. You just need to think and see where you spend money that could be compromised and to be spent on a textbook. I'm not sure your particular financial condition. Maybe you are really in a situation where you are already cutting costs on most of the other non-essential things. But majority of the students spend money on, for example, buying a very expensive smartphone. Instead, you can have the right one that you need, and you could spend the money on buying books. So, can you send? Okay. So, what is the question? Basophil secrete heparin. So that question is not actually from developmental biology. So that you need to look at. I'm not sure whether basophils secrete heparin. That you need to look up. So again, another question on the transcript. But what is the question? In lecture notes tab, there is only slides. I said that in general, based on my experience with other courses regarding transcripts. Okay, I see. So you look through these transcripts, they'll be quite easy. So yeah, so you look through this and then we'll see. Someone is saying boycott NPTEL. Why? Can you explain? Abhishek. So anyways, you're not answering why, but I'll try to justify. See, NPTEL brings you latest possible instructional methods and topics accessible to you at your place free of cost. Okay, so you need to keep that in mind, and you need to realize the people who write textbooks, they really do whole lot of work, often compromising their research and their family interactions to bring out a textbook, and they usually do not make much money out of it. They just get the pleasure of having written a book, and that is all the recognition. So somebody has to do the work. So you just can't expect things free. You need to understand that. When you graduate and go and work, will you do any work free? Someone has to pay you salary, right? So keep that in mind. So I'm sure I have made my point clear, and we'll move on to other questions. So Sudeep, warm greetings, sir. When chimeric cell enter to watch fate map of a particular cell, the model organism do respond it, and how immunological suppression occurs. Thank you. Okay, so you are talking about chimera when we do chimera, like in fate mapping experiments, and how they are not affected by the immune response of the host. In essence, what you are saying is you are trying why is no tissue rejection. This is primarily because we do it at an embryonic stage. So the cell provided there is in the very, very early stage, and due to that, what you have is it is recognized as self, and you have immunological memory for them. At that stage, there is no immune system in the very early stage where we are doing this chimera, and that is why there is no rejection. I guess Gyan is agreeing with me regarding the NPTEL and the copyright issue of the books, and thank you for that. So any other questions? We still have time. Okay. Amit, you are amazing, sir. Your method of teaching is inspiring. Thank you very much. See, this is the reward I get for doing it. But be honest in providing criticism as well, because that will help me to teach better. So criticism from students, like useful feedback, instead of saying boycott NPTEL, will be useful in finding out where we need to improve and where we need to modify or revise, and that also helps. So thank you for that. The binding of microRNAs and their associated risks to the 3' UTR can regulate translation by, in this assignment question, Y is 1, blocking translation initiation, correct. Okay, I'll finish that. And she's saying, and preventing the binding of transcription initiation factors, wrong option. So when they bind to the 3' UTR, okay, so that means already mRNA is made. So now you have regulation only at the level of translation. So most of the time, among the options, I'm assuming it is all about translation. So the transcription initiation factor binding can happen only when the nuclear microRNA goes and binds to the upstream sequence and promotes its methylation. So that is how transcription initiation factor binding would be affected. So, but that is not by binding to the 3' UTR. Okay, you need to focus on the 3' UTR. Here we are talking about regulation happening in the cytoplasm, right? So that's the main point of this question. This question is testing whether you are able to comprehend that. So here the mRNA has come out as a processed mature mRNA, and it is in the cytoplasm. So now the microRNA will only be able to regulate at the translation level, premature release of the peptide, or targeting the mRNA for degradation. It's all post-transcriptional regulation. So for the option that you are thinking, preventing the binding of transcription, initiation factor, that has to be in the nucleus. That has nothing to do with 3' UTR binding. I hope you are convinced. So some other question came. Regret is stronger than gratitude. Okay, so I didn't get that, but anyway. Will it be helpful for my UPSC mains exam? I am sure that's a very good question, but I have absolutely no idea about what they expect in UPSC exams. I am not sure what is covered in UPSC syllabus and what they expect. So you will have to check that. Okay, Harshit got the answer, good. Any other questions? Just feel free to just type it out there. You don't need to feel hesitant, because any question is worth asking, and there should be no, you should be shy to ask a question. Okay, so that's the most important thing. Any question that comes to your mind is a question worth asking. Alright, one more question. I aspire to become an evolutionary biologist with developmental biology as one of my interests. I am also interested in taxonomy and paleontology, which can be related easily. Excellent. You will have a very fruitful and enjoyable career, because I hardly find people with this combination of interest. And the evolution, the theory of evolution and its teachings are actually the unifying theme of biology. And one of the mechanisms to explain evolution is provided by developmental biology. See, evolution operates by selecting among the variants that exist in a population, and those variants in population are actually variations in development. Okay, so that is why evolution and developmental biology are very intimately connected, leading to a field called evolutionary developmental biology, or very affectionately called EODO. That's very fascinating, and that is what I am also interested very much philosophically, although in the lab I work on pure developmental biology problem. But as Ernst Mayr once said, no biology problem gets solved unless otherwise you understand the evolutionary cause of that. So your interests are excellent, and try to pursue that. Do not get dissuaded by anybody else. Any advice you have for me how to combine these fields? So I'm not sure at what stage you are in your career. Okay, this is not like a regular classroom where you are a third year BTech student or whatever. I'm not sure about what stage you are in your career. If you are an undergraduate student, my advice would be, try to choose a lab for your PhD where someone is addressing a question come in the area of evolutionary developmental biology. If you are unable to find that kind of a combination, I would say join a developmental biology lab, and there you can hone your skills on developmental biology, and then you can move on to postdoctoral level to a lab that works on a EO-DO problem. So that is the direction I'll go. And there are a lot of scholarships available. So now itself you look for those. Okay, so your focus is to look for a good lab that works on evolutionary developmental biology of a question that interests you. Okay, so do homework on that by searching through that. Someone has, what is the question, sir? Nervous system. I don't get it. You just mentioned nervous system, but or do. I don't know what that means. I didn't get your question. Can you rephrase your question, Chetan? Mathematical biology fields. I mean, well, see, I will tell you in general. Biology, if you look at it, it is in certain ways very distinct from the way we approach research in other areas. For example, if you take physics or chemistry, where the fundamental questions all have been answered already. So most of the current work in those areas are application driven. Okay, meaning they are invention type of science. Whereas in biology, we haven't understood everything. To give you an example, I'm sure you would be watching news, and yesterday they announced, Emmanuel Carpentier and Jennifer Doudna won Nobel Prize for CRISPR-Cas9. Few years ago, no one knew such a thing exists. Okay, like bacterial cells maintain memory of encountering a bacteriophage in an earlier occasion. Okay, no one thought that is possible. And one of you asked questions on the mRNA. When I was a PhD student, forget about when I was an undergraduate student, when I was a PhD student in the early 90s, no one thought there would be microRNAs. Okay, so there are a lot of fundamental principles still being discovered in biology. You need to remember, by any living organism carries with it 3.5 billion years of experience, and as a result it is an extremely complex system compared to what we study in non-living systems. Due to this, modeling and predicting are not going to be that promising to solve a problem. Okay, so hands-on experiment and discovery is critical here. So in a discovery science, this is my bias, you can call that. Experimental wet lab work is really central, and modeling has only a limited role. Okay, modeling probably can be used for applications from whatever we have already understood. But the way I teach developmental biology and the way I work in developmental biology is not in that direction. We primarily focus on the discovery part, understanding fundamental concepts. So in that sense, biology is more discovery driven and discovery dependent science, and that is the reason it is fascinating. Biology right now is fascinating to us in the same way physics was fascinating to Newton or much later to Einstein. Okay, so biology is at that stage still, and it is extremely complex. Remember, 3.5 billion years of experience is there, and that is why you don't have all the things that are predictable. So that may be a little discouraging, but but that is the fact. Is there any scope as biologist as an engineer? So are you asking, can a biologist work as an engineer, or you are asking whether an engineer has role in biology? I am not getting that clear. So while I'm reading other questions, you can go and reword your question, Ayush. So Ashutosh, please tell us current research about cardiac regeneration using stem cell therapy. See, stem cell therapy, some of the doctors attempt without really understanding how a given stem cell is going to differentiate into a particular tissue. For example, here if you're talking about heart, how a stem cell differentiate, what is the developmental pathway for a pluripotent cell to become a cardiac myocyte, and how that cardiac myocyte gets integrated in the morphogenesis of a heart tissue. There are people who work on the developmental biology problem, but at the same time in India, some of the doctors just go ahead and inject the stem cells and see whether it helps. And if it works, they think that the stem cell therapy works. So I would urge you not to think too much about stem cell therapy right now. The real science-based stem cell therapy is still in the making. It is still very nascent. People try to make simpler things like the insulin producing beta cells in the pancreas, and there it is a lot easier to do to an adult patient, but even there it has not been fully worked out. It is still in the stage of research. Harshit, another question. Can you explain microRNAs and also something about junk DNA? Can you explain the difference between these two briefly and what they are? Okay, another good question. So, 20 years ago, people talked about junk DNA. For example, when I was at your age, my teachers taught about 95% of the genome being junk DNA. But now we no longer think so, and we no longer call any part of our genome as junk DNA. Okay, just erase that word out of your memory and in the concepts of biology. Most of the junk DNA that people thought as junk is where you have the coding sequences for the microRNAs. And then there are a lot of higher order regulations. You know, when you think of gene expression regulation, you're primarily thinking transcription factor binding, etc. That is at a very reductionistic way of thinking about gene expression. There are higher order regulation at the level of chromatin architecture itself. Okay, certain regions of chromatin are coordinately activated or inactivated, and for that, and as well as for making small RNA molecules and the functions of many of them, we have not even understood in simpler organisms like C. elegans that we work in our lab. And because of that, we don't want to call a part of the chromosome as junk simply because we haven't discovered a function for that. So do not worry about junk. There is nothing junk about DNA. Some of the portion originally thought as junk DNA encode microRNA, and others may be involved in the higher order chromatin regulation, and for some we may not even still know the functions. So that is my answer to your last part of the question there. Sudeep, I am pursuing BSc Zoology. I am interested in cell and molecular biology along with evolution. Which course should I pursue in future? Well, you can continue to MSc Zoology or you can go to MSc Biochemistry. Based on my experience with the botany and zoology students, coming from Indian university system, based on the curriculum we have, what I would urge you is, focus on the zoology concepts. Do not ignore them; they are important. But at the same time, build quantitative aptitude. Okay, a flavor or interest for chemistry and principles of chemistry. Okay, that is very important to build a good career in biology. So, principles of fundamental chemistry, you don't need to know very high-fi MSc chemistry student level chemistry. You need to have a quantitative aptitude. Okay, why do I need to quantitate? Why concentrations matter? Because biological reactions are chemical reactions. At the same time, they are not pure chemistry. Principles of genetics are important. So biochemistry, genetics, cell biology, these three are pillars of all branches of biology. But you need to know the biological process. For example, C. elegans is a good organism to look at the neuronal circuitry. To understand that, you need to know what C. elegans is, and that is where your basics of zoology are important. But what I find often is our Indian botany zoology students do not worry so much about biochemistry. So I would advise you to focus on biochemistry. But if a biochemistry student talks to me, I'll tell them to go and learn taxonomy and anatomy and physiology. Okay, so you need to do this. It doesn't matter which MSc you pursue. The point is you need to know these three subjects really well. If your syllabus doesn't cover, take a good textbook and educate yourself. So Radha Dayanidhi kindly enriches with transcription memory, not able to understand the concept. See, transcription memory primarily works through epigenetic modification, if you want to call, primarily because here the modification is not in the DNA sequence. Okay, so any change in the DNA sequence and a phenotype resulting from that is the central concept of genetics. And when there is something transmitted from one generation to another generation, but that is not in the DNA sequence, you call that as epigenetics, and this transcription memory comes from that. Okay, so you could have that in two ways. One, DNA methylation itself, and this DNA methylation can be passed on to the next generation. We have discussed about the CPG islands, how CG being adjacent to each other in one strand would mean GC in the opposite strand. So originally, if the C residues in both strands are methylated, and during replication one of them will carry the methyl group, that helps in perpetuating. So that is one way of transcription memory. The other one is modifications to the histones, that also can be passed on. Sometimes the small RNA molecules, microRNAs, also passed through oocytes, and some very rare things even passed via sperm. And that is how the memory goes from one generation to the next. And I'm not sure whether you are talking about transgenerational transcription memory, or you are talking about transcription memory in one given generation, that is during the adulthood. So what I answered so far is the transgenerational transcription memory. If you are asking about one generation, then it is primarily through the polycomb and trithorax proteins that bind to the chromatin and modify the chromatin itself, that is by methylation and acetylation of the histone residues. And that modification remains part of the chromatin. You don't need a transcription factor to come and keep activating. And that is why it is memory, meaning it is a continued state of activation or continued state of inactivation, and that is done by these modifications to the chromatin, so that the transcription factor can readily access or it cannot access. And Kiran, during development of embryos, is there any reason why brain develops first? Not necessarily. Brain doesn't develop first. The first thing that is set apart is actually the germ cells. Okay, they are the ones that are first set apart. Then you have the rest of the things forming. There isn't any specific reason why brain develops first. Anurag, my question is, does capping and tailing sequences also get translated and they code for any protein? No. Capping is actually a single group, G guanosine residue, that guanine, that is trimethylated. So that is not a triplet codon. Tailing is poly A tail. Okay, so A residues only are added, and that again is not translated. Just your stop codon comes before that. Harshit again. Is there a possibility to do an NPTEL internship under you, provided all requirements met after this course? If yes, then what can be done and expected? I have to check with the NPTEL. I'm not sure NPTEL internship how it works. But you can, once this COVID lockdown is open and the campus is back to normal, you can apply to our lab for an internship. There are two routes to do that. One, IIT Madras offers summer research fellowship. Look for that advertisement and apply through that. And at that time, you email me also. Second one is Indian Academy of Sciences offer a summer research fellowship. You check their website and apply there, and when you apply there, you express your interest to work in our lab. And now that you have asked so many questions and specifically expressed your interest, I'll remember it and then look at the application accordingly. Some questions asked in assignment, though we answer the same, what do you have told in video lecture getting incorrect? Why? You need to be specific about a question. So you can shoot this question on the discussion forum and the TAs will answer you, and when they have problem they will discuss with me and answer it, because right now I'm not sure which question you are talking about. You need to specifically address an assignment, and there are other students who have done that, and the one or two questions even have been corrected and I have instructed them to re-evaluate. So what you do is, in the discussion forum with the TAs, you raise a question about a specific question, then only we will be able to satisfactorily address your concern. Preeti, why clonal propagation of C. elegans is easy? It is because it is an hermaphrodite. It's an interesting hermaphrodite, okay? It, like us, meaning human, it has a diploid germ cell that undergoes meiosis, meaning homologous chromosomes pair, crossover happens, normal meiosis happens, and then you get haploid gametes. And you have sperm and egg, fusion of which raises the second generation, next generation, subsequent generation. So it's a sexually reproducing organism. The difference is, to put in a very simple informal language, a hermaphrodite of C. elegans equals a human female who is capable of producing a few sperm. Okay, assume that a human female during adolescence, the first few germ cells that mature, meaning undergoing meiosis, becomes a sperm, and the sperm are stored. And then subsequent germ cells that undergo meiosis enter oogenesis and they make oocytes. And these oocytes use the stored sperm to fertilize. So a single worm can actually produce an embryo without requiring to mate with a male. And that is why clonal propagation is easy. You take a C. elegans before at any stage and put in individual separate petri dish on an E. coli lawn, it will, since it has sperm and egg both, it will reproduce. And that is why clonal propagation is easy. Can embryo editing be done using CRISPR? Yes, people are doing it. So we do it routinely in our lab on C. elegans. We have already generated edited genes and published papers, and you can look up at the website. If you search with my name, you will be able to find a paper. I'm not sure whether it is there in the previous paper. The paper published in development, there we have used. And that is at the embryo level, actually even earlier level. So in C. elegans, the editing happens by injecting into the germline, and the gametes themselves are edited, and editing can happen during embryogenesis also. And in organisms like Drosophila, the molecules are injected into the embryo and editing is done in the embryo. So Abhilasha, what are skills that zoology students should know which can help in research area? Learn biochemistry, genetics, cell biology thoroughly. Okay, the basic concepts. You don't need to go into very advanced state. Okay, no one will find fault with you if you didn't know what CRISPR is, what miRNA is. But people will not be happy if you did not know Mendel's laws. Okay, you need to thoroughly and conceptually understand what does law of segregation means, what law of independent assortment means, or why do biologists in the lab use molar term for concentrations and not percentage terms, right? If I make glucose solution, I can always say 1% solution. That is precisely doable in any part of the world, 1 gram in 100 ml. But why do we make 1 molar solution? What is pKa, what is pH? Like this, fundamental concepts of subjects are important, and such fundamental concepts of genetics, biochemistry, cell biology are the key, and curiosity. If you have this, then you are well equipped. And I am sure you are already learning zoology well. Do not belittle taxonomy. Taxonomy is very important, and that gives you an idea of the evolutionary history of organisms. Okay, do not badmouth or look down upon taxonomy. That would take care of your interest in research. Someone asked CRISPR-Cas9 questions. I'm not getting what is the question. Which topics are important for CSA or NED? I don't know. You look at their syllabus. I would say if you have the fundamental concepts clear, you should be able to take any exams. Unable to understand DNA methylation, which you taught in week 2. What is your specific question on methylation? Do you believe in the RNA hypothesis? I assume you mean RNA first. Yes, that is the prevailing understanding in biology. There is nothing believing and not believing in science. Based on our current evidences and whatever our current understanding of the chemistry and biology of DNA and RNA, it seems reasonable that RNA must have come first, because RNA has the capability to be a catalyst as well as an information storing molecule. Remember, in science, scientific temper is the key. We do not worry about faith and belief. We go by arguments based, reasoned out from known facts. Okay, and therefore it is subject to revision as well when new evidences come. Just wanted to thank you for providing a course in developmental biology. Thank you very much. I'm not pursuing for the certificate but for knowledge. I'm sure this will be helpful for GATE paper too. Yeah, I'm sure, but GATE syllabus has many other things and you need to cover them as well. So again, one more question on the same, so the same answer holds good. Unable to get gel mobility assay. Please explain. Okay. See, the term itself tells gel mobility shift assay. You are missing that one important word there. So when you run a gel, let us say a polyacrylamide gel of shorter nucleic acid molecules, let us say a small piece of DNA, like about 20 base pairs or 20 nucleotide long single-stranded DNA. It will run at a certain rate. Now, if a protein is bound to it, then it will run at a slower rate than without the protein. And that shift in mobility, okay, meaning when you look at the gel, you see the band with the lane containing the protein is shifted up, meaning it is moving slowly compared to the free one. That is why we call it as mobility shift. And that retardation in the mobility indicates that it has bound a DNA molecule. So it helps in understanding whether a given DNA and protein, whether they interact or not. So that is what is gel mobility shift assay or gel retardation assay. retarding the mobility of the DNA. So the main point here is a short DNA oligonucleotide moves slower on a gel when bound to a protein. This reduction in the mobility rate is diagnostic of its interaction with the protein. If it does not interact with the protein, then it is not going to be slowed down. Then you know that protein doesn't interact. And to make sure whether it is non-specific interaction or specific interaction, you can have excess of a non-specific DNA of the same length. Now, if the protein was non-specifically binding, then that excess non-specific sequence, meaning non-specific in the sense not related to your sequence of interest, that will compete out the binding of your sequence to the protein. But if the protein specifically bound to your sequence, whether the non-specific molecules are there in excess or not, that will not affect the retardation by your oligonucleotide. So that is how you can find whether the protein binds to the given oligonucleotide or not, and does it bind specifically or not. Both can be tested using gel mobility shift assay. An organism is exposed to environmental pollutant. Epigenetics is a possible mechanism presumed. The modified basis if carried to generation for how many generations? So if the bases are modified, it is forever. It is subject to selection later. Okay, it could be permanent. But if it is epigenetic modification, like modification to the chromatin or methylation kind of modification to the bases, then as long as the methyl transferases work, it will be there. But for long, but it's very difficult to say how many generations. It is very specific to give a specific number there. Okay, it depends on the selection forces that operate consequently. So one more question from Ayush. Going through the course, I believe developmental biology require a sound prior knowledge of molecular biology. Yes, and in addition, a sound knowledge of genetics also will be good, and cell biology and biochemistry will be helpful. But you can learn them as you go along. Yes, that's correct about RNA hypothesis. See, the history of life started with simple molecules that formed under the primordial earth, based on the conditions that existed, self-replicating molecules and a way of metabolism in which probably became possible in hydrothermal vents in deep ocean, and from there life began is the current understanding. And in that process, people believe RNA molecules would have been such self-replicating early molecules, and that once when lipid molecules were available, because lipid molecules readily form bilayer structures, and when such lipids enclosed or encapsulated these RNA molecules, that led to the formation of a protocell. I would urge you to read the relevant chapters in a book called Biology by Campbell. Okay, so if it's a very famous, probably the most famous biology textbook. There it is all explained in very good detail and very easy to read. Is alternate splicing subclass of differential splicing? Well, differential splicing is a form of alternate splicing. So when alternate splicing happens, different forms, like for example, let us say a gene has exons 1, 2, 3 and 4. In one particular tissue, if exon 2 is skipped, 1, 3, 4 are joined, and in another tissue 2 is not skipped. That tissue difference is what you call as differential splicing. Okay, so alternate splicing means these two forms like 1 2 3 4 versus 1 3 4 are alternate splicing, and when it happens differently in different tissues you call differential splicing. So okay, I'm sure you all enjoyed. I will take more questions during our next discussion meeting, probably in a month's time. Until then, enjoy learning developmental biology, focus more on the learning experience and learning the concepts. That will automatically take care of the exams. Do not read only with the exam in mind. All the best.
Feedback.
Oh. ரொம்ப டிலே இருக்கு. எப்போ நான் பேசுறது இப்ப தெரியுதாங்க என்ன